Questions can be typed in the chat box, or I think Martin, people can just shout out?
Yeah, we've gone less formal these days, so people can, if we wait to the end of the presentation, then Barbara, it's up to you, however you want to manage it. You can read out people's questions or whatever, it's fine.
I know Aldebaran will deal with them if they come as they go, and then the talk is being recorded. So,
right.
Okay, so I'm going to this slide. So Aldebaran got her PhD at the University of California in Berkeley with Terry Machen, and she did postdoctoral work first in Germany with Irene Schultz, and then in Italy with Tullio Pozzan. And from there, she joined the faculty of Harvard Medical School and has been there since. Many of us remember that Aldebaran was an organizer and a co-organizer of 2 FACEP conferences on calcium and cell function. My first introduction to Aldebaran's work was with her elegant measurements of free calcium in subcellular compartments. And I think this set the stage for the future work. Now her present work is focused on the reciprocal interactions between cyclic AMP and calcium signaling regulation. And her expertise in monitoring calcium in very small organelles from this early work has led her to optimize methods to directly visualize cyclic AMP signaling in the primary cilia. And with that, she can address the function of signaling in this small organelle. This image here, which I think is also, and you're going to see again, shows cells expressing a cyclic AMP reporter targeted to the primary cilia, and that's in green. And you see this extremely small, I don't know if you can see my pointer, this extremely small vertical compartment. And this was with the serotonin receptor marker, and also, cyclic AMP. And this image illustrates the geometry and the relatively small size of the cilium with respect to the rest of the cell, and the near perfect co-localization of these cell components. And today she's going to discuss her current work examining this organelle, and the title of her talk is GPCR, or G protein coupled receptor signaling in the primary cilia. And I'm really looking forward to it, and take it away, Aldebaran. I'm going to stop sharing.
Wow, thank you very much, Barbara. That's really nice. Let me start sharing now. My entire screen. And here we go. Hopefully it's coming out. Something. Do you see my screen? Because I don't see it.
So Aldebaran, you need to go to your presentation.
There we go.
That's it. And
okay, here we go.
It
takes a little slow. All right, so I can't see myself, which is a little disconcerting, but we'll deal with that. And I can't see the rest of you either, just so you know. So you could make bad faces, whatever. So Barbara, thank you so much for stepping up and introducing our talks today. There'll be 2 of us today, myself and Matt Strobel, who has just recently got his PhD, and I'll introduce him at the end of my presentation. And so, yeah, as Barbara already alluded to, I'll be talking about these little things here, the primary cilium. And so what you're looking at here is a field of IMCD3 cells with the ciliary marker, and you can see that these little tiny organelles are sticking up out of the cell into the bath. Okay. So, and this is what I'm going to be telling you about. So cilia are non-motile, but they do move around, just passively due to different sort of thermal movements in the bath and whatnot. And they have this microtubule core made of acetylated alpha tubulin, and so there are these doublets of tubulin. And they're assembled from the mother centriole. So they sprout up from the basal body. And there are these important proteins, the intraflagellar transport proteins, that are involved in the assembly of this structure. So without these, you can't have a cilium. And some other important things is that you may not realize that they are present on pretty much all vertebrate cells that are in the non-dividing state. They're non-motile, as I said. And there are about 1,000 proteins that make up a primary cilium. And so this is the primary ciliome. So, and many of these are distinct proteins. Okay. So cilia are most known for their role in signaling. So they're essential for hedgehog signaling and also very important for a couple of other pathways, notably Wnt, planar cell polarity, Hippo, and then I could also mention Notch or TGF-beta signaling as well. So just a little bit more about cilia before I start talking about the main part of my talk. So David Clapham had some really nice work on the primary cilia some years back. And one of the things that he showed was that they have an independent membrane potential, and they also have an elevated resting calcium concentration. And this is probably because they have a number of calcium conducting channels in their membrane that sort of allow a leak of calcium in, yet they don't have calcium extrusion mechanisms to my knowledge. So calcium inside the cilium might be around 600 nanomolar. They also have this unique lipid composition, and it's mostly PI4P, but with low cholesterol. And then the other thing to remember about cilia is that they have this interesting barrier at the base. So it's this made up by this thing called the ciliary necklace and some other proteins, and it's sort of reminiscent of the nuclear pore or an axon initial segment. So it's a sort of semi-selective for smallish proteins. Okay, so one thing you may be interested to know is that cilia, we can measure calcium in cilia, and what we're looking at here is just sort of the passive accumulation of ciliary calcium according to what's going on in the cytosol. So we're just looking here at some IMCD3 cells that are undergoing some spontaneous calcium oscillations in the cytosol, but we're able to measure that with different sensors. For example, this one that we got from UBin. All right, so that's sort of my big homage to calcium, except that I do want to say for the stimophiles that sadly there is not stim in the cilium, and to my knowledge, nor is there ori. It's an organelle, so it doesn't have other organelles like the ER inside of it. But here's just stim, the distribution of stim YFP before and after, and then this is the cilium that's labeled with this marker. Okay, all right. So they don't have stim, they don't have ori, but what they do have that was really interesting to me as a person who studies crosstalk between calcium and cyclic AMP is that they have adenylyl cyclases, many of them regulated by calcium, especially AC5 and 6. And they also have a very interesting complement of G protein coupled receptors. And these are almost invariably linked to cyclic AMP, either through G alpha S or through G alpha I. To my knowledge, there are not really any documented instances of other receptors linked to G alpha Q or calcium signaling. Okay, so in addition to those receptors, they also have G alpha S, which is very abundantly expressed. And so they have all the machinery here to make a cyclic AMP signal within the cilium. Moreover, they apparently have phosphodiesterases to terminate cyclic AMP signals, and they have effectors for cyclic AMP. Particularly, they have the pKa holoenzyme. And finally, they also have many ciliary pKa substrates. And some of these would include polycystins like polycystin 2, PKD2. And very importantly, they have transcription factors such as Gli transcription factors that are mediating the Hedgehog pathway. Okay, so all of this leads us to ask whether this is something like a little privileged cyclic AMP signaling microdomain. So do these receptors, when activated, do they produce cyclic AMP? And today I'm going to be telling you about these receptors marked in red, mostly the 5-HT6 serotonin receptor, D1R dopamine. This is another receptor involved in Hedgehog, and then these two receptors that are G-alpha-I coupled, somatostatin receptor and the smoothened receptor. So in order to probe this little microdomain, Jason Zhang in the lab and Jeff Falcone, along with Silvana Corci, we tested many different variants of cyclic AMP sensor and came up with this one that seemed to work the best for us. We used an ARL13B targeting sequence or dopamine 1 or 5-HT6 receptor, and those were fused with this FRET-based sensor from KCL-Link. And the nice thing about this particular probe is that we can use it either in FRET mode, which is ratiometric, or we can have this sort of dimensionless, it's not really dimensionless, but we can use FLIM, so fluorescence lifetime imaging, to look at the lifetime of the donor, so the M turquoise in this sensor. And this turns out to be kind of nice because it's independent of the intensity of the sensor. All right, so we can image in 3D the FLIM, the lifetime, and so here these are cilia with very well targeted sensor. Okay, that's been targeted through this 5-HT6 serotonin receptor. Or we're sometimes using sort of a more gentle illumination with FRET, okay, in epifluorescence. And here you can see the response to forskolin in these cells. And one thing that you'll notice is that there's a lot of non-targeted sensor and also targeted sensor in this particular cell. Okay, so here I just show you an example of a FRET experiment in which we've got both targeted and non-targeted sensor, and then we've stimulated the cells with 50 picomolar AVP. So these cells are expressing V2R, the vasopressin receptor, and this FRET reporter for cilia. And so we're able to measure in both compartments simultaneously in this case. And one thing that we found out is that in contrast to some prior reports in the literature, when we're overexpressing V2R, we're not really finding good evidence that it's in the cilium. And so what we conclude is that this cyclic AMP that's produced out in the cell body is traveling freely into the cilium. And this is probably no surprise because calcium is moving freely into the cilium. So why wouldn't a small molecule like cyclic AMP move there? Okay, so another thing we found, and these data are published, by the way, so I'm just going to go through really quickly and tell you what the highlights were of this paper that we had a few years ago. The other thing that we discovered is that the resting ciliary cyclic AMP concentration is not really different from that in the cytosol. So both cytosol and cilium maintain a resting concentration that's in the submicromolar level. All right, and so here what we've done is we've permeabilized cells with digitonin because the cilium doesn't have much cholesterol. We can sort of selectively permeabilize the plasma membrane and then do an in situ calibration. And these results were confirmed when we looked at FLIM. So this FLIM lifetime is going to be reflecting cyclic AMP. And here are two cells, one that has perfectly targeted sensor in the cilium, and here's one with sensor in the cell body and cilium. And actually, if anything, the lifetime is generally a little bit lower in the cell body than it is in the cilium than it is in the cell body. Okay, so so why is that interesting? You might think, well, that's boring, but because of course, cyclic EMP, it seems like it should be the same in the cilium and cell body. But it sort of contradicts this prevailing view in the literature that cilia maintain a high cyclic EMP concentration of around 5 micromolar. And that's due to the constitutive activity of this G protein coupled receptor, GPR161, that's always working, always making cyclic EMP. And the functional importance of that would be that you're getting continuous activation of protein kinase A and activation of GLI transcription factors that are involved in repressing the Hedgehog signaling pathway. And there's also just sticking calcium in here again for the benefit of everybody. There's also some suggestions in the literature that calcium entry through channels might inhibit this process and then thereby relieve this inhibition. But, unfortunately, we didn't find that there is high cyclic EMP in the cilium. And so I think the explanation for this pathway here at rest, this constitutive repression of Hedgehog is something a little bit more complicated. So just quickly, I'll tell you what we found when we tried to look at receptor function in the primary cilium. So here we've got, again, this 5-HT6 receptor linked directly to our FRET reporter. And what was interesting to us is that whenever we had perfectly targeted 5-HT6 FRET reporter, we were unable to resolve any kind of response to serotonin. So here we're putting this relatively large dose of 5-HT, and we're always comparing our response to what happens after forskolin plus IBMX, and we really could never see anything. This is in contrast to what happened after we stimulated the Hedgehog pathway, however, where we started to pick up very consistent responses to serotonin in the cilium. And so in this case, what we're doing is we're stimulating the Hedgehog pathway with this agonist SAG. It's called smoothened agonist. And I think at this point, I should probably tell you a little bit about SAG and how it's working and the Hedgehog signaling pathway, since it's so much related to cyclic EMP signaling in the cilium. So we'll start with the off state. So this is the most abbreviated version of Hedgehog signaling you'll ever see. So what happens is we have this big 12 transmembrane receptor called patched that's in the cilium, maybe also in the plasma membrane, and it's repressing another receptor called smoothened, which is the target of this SAG. And then we have the idea that the GPR161 is constitutively phosphorylating, although apparently not with an elevated cyclic EMP level, phosphorylating these GLI transcription factors that then move out, and they are repressing Hedgehog target genes. So this is the off state. In the on state, we get Hedgehog ligand, which is secreted from nearby cells, engaging with the patched receptor, which moves out of the cilium. Then the smoothened receptor moves into the cilium. And we also have the exit of GPR161 and these GLI transcription factors, GLI1, 2, that then are able to go to the nucleus and activate Hedgehog target genes. So when we treat with SAG, we're bypassing that whole thing I just showed you of many receptors moving in and out of the cilium. And actually not just receptors, a lot of things are moving in and out of the cilium when we stimulate the Hedgehog pathway. And so we can put SAG on, and this will result in translocation of smoothened to the cilium and the direct activation of Hedgehog target genes. Okay. So there's also another compound, cyclopamine, which is an antagonist of this whole process. All right, so just remembering that, then I just point out that we looked more thoroughly into some different cell models to look at what happens to the GPCR function when we activate with SAG. And so we can try different cell types. This is just, I'm going through this quickly. So 3T3 cells, they actually give a small response to serotonin, but it's greatly amplified after we treat with SAG. Actually, interestingly, nothing much happens with cyclopamine. We can also visualize the antagonistic action of this G-alpha-I coupled receptor, somatostatin receptor 3, and you can see that here. Okay, and we can also do similar experiments looking at the dopamine receptor D1R. Okay, and get similar results. So an amplification of the response. Okay, so what I've showed you, just summarizing these previously published data, is that primary cilia, they appear to host this independent local cyclic AMP signaling circuit. But so this is the local cyclic AMP production. And I'll point out too something interesting, which is that you won't detect cyclic AMP produced here out in the cytosol because it's just sort of diluted out in that compartment. So this is why we don't see much. And so also these receptors, at least these two that we looked at in the cell models that we looked at, they are enhanced after Hedgehog pathway activation. And so what this might suggest to us is that we have this sort of oblique way to influence, let's say, Hedgehog pathway that doesn't involve GPR161, which is an orphan receptor. We don't know what the ligands are for it. So these other GPCRs might be influencing these other pathways. And I'll just say that these GLI transcription factors are just one of many potential targets for PKA within the cilium. Okay. So now our question is, why is it that we couldn't see any sort of activation of the serotonin receptor in those cells once they were in the cilium? And so one possibility is that they have switched their affinity for their G-alpha subunit. So many receptors, the calcium sensing receptor and others, are known to couple promiscuously to different G-alpha subunits. And there's also some evidence that the bile acid receptor TGR5, this is from Nicholas LaRusso's group, that TGR5, once it goes into the cilium, can switch its affinity for its G-alpha, its usual G-alpha binding partner. So we thought, and then also I'll mention too, if any of you saw Shushen Shu's very beautiful presentation at the FASIB calcium meeting, there's also some idea that the 5-HT6 receptor, once it gets into the cilium, may have some, not atypical coupling. And so we turn to these really beautiful little tools here, which are the mini G probes that we got from Nevin Lambert. And so what these are is they are little constructs that they have sort of all the essential elements of a G-alpha subunit in that they can couple in place of the regular G-alpha subunit that's there endogenously and substitute for it. And but this only happens when this G protein coupled receptor is in its ligand activated form. So for example, if we have the serotonin receptor and it's has been activated by serotonin and is competent to couple to G-alpha S, we will see a movement of these mini G proteins towards the membrane where it binds to the GPCR. And so it turns out that these work really well for cilia. And so what I'm showing you here is actually that we can visualize in the cilia the activated form of smoothened. So what we have here is M-cherry smoothened, and we've treated these cells with SAG for about 10 minutes. And then it's very clear before we don't see anything, but then we see this movement of this mini GI, because smoothen's a G-alpha I coupled receptor, into the cilium. And so this is a great way to look at the activation of this receptor to show that it's been activated. And what was really gratifying for us is that we can also look at endogenous smoothened. Apparently, this is what we're looking at, because we haven't co-transfected these cells with the smoothened receptor yet. Around 10 minutes after we treat with SAG, we can see the movement of the mini G venus into the cilium. Okay, so that's nice. And then we wanted to know is M-cherry 5-HT6, is the 5-HT6 receptor able to couple to G-alpha S? And so we used the version of the mini G, which is specific for G-alpha S. And what you can see here is that before, here's our 5-HT6 labeled cilium with a functional receptor. Before serotonin, nothing happens, but after serotonin, we get really nice accumulation of this mini G construct. So we used mini GQ, mini G12, and mini GI. I just show you the result from mini GQ because all these results were negative with these other constructs. So what we conclude is that there's not an alternative coupling between 5-HT6 and its G-alpha subunit once it's moving to the cilium. So that's not our explanation. So these are some of our newer unpublished data. And then I would like to just conclude by talking about what some of the remaining questions are for us. So why does Hedgehog signaling amplify ciliary GPR signaling? So there are a lot of things, as I mentioned, moving in and out of the cilium upon Hedgehog's treatment. And we think that some of these things might provide some clues for us. There have been some very nice papers on this, for example, from David Mick's group, who did a really beautiful proteomic study and showed what's moving in and out. And one possibility is that since the pKa regulatory subunit is moving in and out of the cilium, what we might be looking at is a sort of buffering effect. So under control conditions, there's a lot of regulatory subunit present, and this seems to disappear after stimulation with Hedgehog. And then the other big question is, what does cyclic AMP do once it's in the cilium? What are the targets that are being regulated, and what are the processes that are being affected? And to this end, we've been working on creating a ciliary cyclic AMP toolbox. So to consider this idea about pKa regulatory subunits buffering cyclic AMP in the cilium, we have a construct that's kind of similar. It's based on the pKa regulatory subunit, and we're calling that cyclic AMP sponge. And we've also created constructs that are targeted cilia to inhibit protein kinase A, and as well as activate well, it is protein kinase A targeted to the cilium. And something that we've also been working on quite a bit is this particular phosphodiesterase isoform that we've targeted to the cilium, and this looks pretty promising. And I think this question is important, and we're kind of on the right track. So just last year, a very beautiful paper came out from Melissa Truong from Jeremy Ryder's lab, where they created some other kinds of tools, and they looked at, for example, zebrafish development, and showed that it was very much influenced by this local signal within the cilium. Okay, so now I'd like to sort of end by giving a little bit of introduction to Matt Strobel's talk, which will be following mine. And he's going to be talking about cilia in neurons. And so here we've got let's see, this was ARL13B TD tomato. And again, maybe some of you saw this really great talk by Shu-Shen Xu. And he was showing, he provided some pretty good evidence that's not published yet, that there are serotonergic axons that are making contact with primary cilia that are expressing the 5-HT6 receptor. So I think this is really, really exciting. And he showed a lot of nice data about the downstream effects, so signaling that was initiated here that was having downstream effects in the nucleus of the neuron. And so I think we have a lot to learn in the future to try to understand what's going on with especially neuronal cilia, where there's so many of these GPCRs, and we have really kind of no clue what they do. And we sort of don't know much about what GPCRs are where in the body, in the cilium, so in our whole bodies. All right, so with that, I will just stop and mention people in my lab. Again, there was Jason, Zhang, Jeff, Falcone, and also the more recent work is done by Danielle Arena and myself, and of course, always Silvana Curci and some other very wonderful collaborators, students that we've had. I'll mention Mao Chen from University of New Hampshire, and Ivana Kuo and Barbara Ehrlich in particular. Okay, so now with that, I'd like to conclude and turn it over to Matt Strobel. So Matt, you could probably start getting your screen going while I introduce you. Are you there?
Yeah, just had to find the mute button. I think you have to cancel your screen though before I jump in.
So let me exit. Oops. All right, good. So it's really my pleasure to introduce Dr. Matt Strobel, who just got his PhD in August from the University of New Hampshire. And he's been working with one of my collaborators, Dr. Mao Chen. And so he studied genetics at the University of New Hampshire, and prior to that, he got his Bachelor of Science in Evolutionary Biology from the University of Connecticut at Storrs. And it's my pleasure to turn it over to Matt, who's going to tell us what cilia do in mice. Okay, thank you, Matt. Take it away.
Hey, they do many things. So the reason Aldebaran and my paths crossed is because she was collaborating with Mao Chen, as she mentioned, and Mao studied adenyl cyclase 3, and more specifically, its link to major depressive disorder. Through several big genome-wide association studies, AC3 has been linked to depression, which is pretty significant because depression affects about 7% of adults in the US, and COVID probably hasn't helped that. But one thing that's unique about adenyl cyclase 3 compared to the other 8 membrane-bound adenyl cyclases is that it's predominantly expressed in neuronal cilia. Here we can see our nice staining of it, and it's really not found strongly outside of the brain. And what my advisor found through his previous research is that when you knock out AC3 in a mouse model, we get a host of depression-like phenotypes. All of these are just behavioral tests where we're just showing they've been impacted in a negative manner. And this kind of backs up. We already had linked AC3 to depression through human studies, and now we kind of have a little evidence that it's playing a similar role in mice, so we know that we can use this as a model. So my role was kind of taking his data and starting to tease backwards. We have some behavioral data on the link between AC3 and depression, but let's start working towards the molecular side. And in addition to our depression phenotypes, they also turn into little fuzzy tennis balls. So one tool we use for this, even though we're studying AC3, we developed an Ift88 knockout. And the reason we did this is AC3 is membrane bound, and there are a few other adenyl cyclases which aren't exclusive to cilia, but sometimes can localize to it, and sometimes can get a compensatory effect. So Ift88 is involved in this binding of cargo to kinesin and is used for, which ultimately is used for extending and building our primary cilia. So by knocking out Ift88, we're disrupting the machinery and we're ablating the whole cilia. We're not giving any adenyl cyclases anywhere to go to compensate or signal from. So it's a bit of a way to give us a strong benchmark for a phenotype. And then we typically duplicate our experiments in AC3 mice to see if it still follows through. And when we were just screening these initial Ift88 mice, one of my lab mates, Yuxin Zhu, found this interesting piece where she saw their memory was impacted. Here we have a paired stimulus test where these mice are in a cage, they hear a loud tone, and shortly after that, they receive a little tiny foot shock. And typically they start to learn, oh, there's the tone, I'm gonna get shocked. They kind of freeze and pause and wait for it. But you can see here in our Ift88 knockout, their learning's a lot more delayed and heavily impacted. And I found this really interesting because primary cilia's claim to fame has primarily been through development, its control of the cell cycle, and also the various developmental pathways. But there's a growing body of evidence showing that primary cilia are important for other neurological processes throughout life, and starting to be linked to a number of more diseases and human conditions, or at least being studied for them. So I found this really interesting, and she was going a different direction for her research, so I wanted to take this and start teasing backwards. Same idea, we have some behavioral data. I wanted to start working back towards the molecular side of it. So the first thing I did with this is ask, well, if we're impacting memory, we're probably impacting our spontaneous neuronal activity. And again, a kind of a new and novel area for primary cilia research. So I did a 24-hour EEG study where I implanted intracranial EEG. Here we can see it. They actually, so this is dental cement, not his brain, even though it's pink. You plug them right into a computer and they run around happy as can be and don't even really pay attention to it, which is kind of interesting. But here we see, so this is a quick snapshot of the data. And I said this is a 3-channel recording. So my first channel, EEG1, that was over the occipital lobe, so visual recognition. My second channel is over the frontal lobe, so more cognition and thinking. In my final one, EMG, that's muscular activity, so if they're moving around and whatnot. And you can see here, you know, I've broken it down into these 3 phases. Are they awake, non-REM, or REM? Which the differences are here nice and obvious. These are 10-second windows that I've picked so we can compare this. When they're awake, they're taking in a lot of visual data. They're looking around. There's a lot of activity on our occipital lobe. When you're awake, your EEG activity and cognition, it's typically a high frequency. So lots of packed together waves in low amplitude, so not much height. And then they're awake and running around, so there's a lot of muscular activity. In non-REM sleep, they're sleeping. There's not much visual cognition happening. Our frontal lobe, it starts to space out. A little bit longer of a wavelength, a little bit taller of an amplitude. You can see we're starting to get this kind of diamond and spindle formation. And typically asleep, not a whole lot of activity. Maybe sometimes you can actually see on here when they're light twitches or something. And REM is a little bit of a combination. No activity on EEG1, Very high activity for EEG2. It's almost like being in an awake mental state, but no EMG. And the first thing I did was break down all the data into these three categories. The reason being, all three of these are associated with different steps of memory and learning and consolidation. So I was able to do this with a mix of automatic and manual scoring. Here's my first go of just cluster scoring, where we can presume if there's a lot of EMG activity, they're probably awake. So I just circle all that data and label it as awake. If there's a lot of EEG activity, that's probably because they're in our non-REM sleep. We've got a lot of power happening there. And then down in the corner, there's not much happening on either. So I went through, did a rough cluster scoring, and then had to go through and manually correct and check everything. And with this data, each of these windows I have, or epochs, are only 10 seconds. I have 24 hours for each mice, which my sample size was originally 10 knockout and 10 control, but I had to eliminate 2 just because some problems with the file corrupted. And because it's so bulky, the first thing I did was just transform it into frequency. I made these power spectrum density plots to look at overall, how's our brainwave power being affected. This way, instead of the x-axis being time and crashing my computer because it goes on for so long, now it's frequency. So we see here going down on the left from our very low frequency, here I've got it cut off at about 1 hertz, all the way up to our high frequency gamma waves up to 100. And we can see right away here, there's a really big jump in awake and a small one in our non-REM and slightly in our REM too. So what we're seeing is reduced peak waveform activity and also a slight shift. And here's a little more raw version, and we just see that reflected in our cumulative probability of the data. That again, in our knockouts, it's completely cut down. We don't see anything over this 200 power mark. And here, you can see our significance. It's only 1 star, but part of that's because there's areas that are more affected and less affected. So I teased this apart and broke down each one of the wavelengths just so we could look at it a little better. And here's what I found. You can see, it's a reconstruction of the previous data. When they're awake, there's a huge difference in power in delta and theta, versus in their sleep states, it's their alpha and beta that are affected. And one thing that's I found interesting about this is it's essentially the background waveforms that are being affected. When these mice are awake, they're primarily using a higher frequency. They're primarily operating on alpha and some beta wavelengths. Delta and theta, less common when you're awake, and those are the ones that were heavily impacted. Versus asleep, it's the mirror image. So when you're asleep, it's predominantly delta and theta waves. Not much alpha and beta happening, but we see the alpha and beta are the ones that are impacted. So I found that interesting that it's our kind of background wave frequencies that are being impacted. And again, when we look at the density of this data, it just illustrates it even more. I mean, for our knockouts, all of our information is pretty much at our 100 power units, versus in our control, it's much more distributed and shifted higher. And this was also cool just to see, because just looking at the raw data, I kind of noticed there's a difference between the wild type and the knockout. And actually seeing those previous results kind of verifies it. Oh, that is true. There really is a reduction in their activity. And this was important to us because we know and we've seen in human depression patients that they also have reduced waveform activity. So we're seeing in mice the same phenotype we're getting in humans. So again, we're starting to validate this model a little more and find out the importance and the role of primary cilia in that. So this was a little more on our general wave activity and a little more on the depression side of my research. The next thing I wanted to look at was getting back to memory and teasing out, again, those previous results that I had showed you. So before I jump into that, we're going to talk about phase amplitude coupling, because that's kind of what I use to look at it. And I already mentioned before, frequency is how many waves we're having in a time period, so the rapid and how occurrent they are, versus amplitude is our height. And those are things a lot of people are fairly familiar with. But now I'm going to delve into phase a little more, because that's more what I'm looking at. And phase is just the timing of these. As we can see, all three of these waves are the same amplitude and the same frequency. They're just offset. So that's 3 different phases, which we can measure in degrees to look at this offset. And for phase amplitude coupling, what it is a harmonization of 2 different wavelengths. So here we can see we've got this low frequency wavelength as well as this high frequency wavelength. And when the peaks of their amplitudes are in phase and nice and lined up, we get this magnification and this harmonization effect that produces these really strong Morlet wavelengths. And when that happens, you get a lot of action potential firing, a lot of rapid spikes, which are marked here as these little red lines. Versus, so here, our peaks are in line and we get a magnification. Here, I haven't impacted the amplitude or frequency at all. I've just shifted them. Their phases are no longer lined up and they kind of cancel each other out. It's almost like kids jumping on a trampoline. You know, you can only jump so high, but if all your friends time it together, they can launch you a lot higher. So that's what we're seeing happening here. And with phase amplitude coupling, we already know it's important for memory formation. That's already been established. And it's important both for new memory formation as well as the long-term potentiation of our memories and holding on to things. So in my case, I did theta waves with our high gamma waves as my low frequency and high frequency. And you can see here when we're getting this harmonization in this long-term potentiation, we're causing an influx of calcium into these synapses. So I'm almost using this experiment as a surrogate for measuring calcium signaling. I actually had built a full electrophysiology lab and started doing single cell patch clamp recordings, but COVID disrupted that whole project and shuffled it around. So I ended up doing working on this experiment and project first. And one way I quantified and showed this was using these polar histograms, where essentially this red line, it's illustrating both a direction and a strength. Because remember how we can measure phase with degrees? This is showing that the phases all had a similar area they were pointing to. A lot of our data is all in this area, as well as the long length means there was a very strong wavelength or waveform here. Compared to if there's no phase amplitude coupling, everything's kind of mixed all over the place and much less concise. So here's my initial results. As we can see here, you know, just as we showed, we have a long red bar here, meaning our mean vector has a pretty decent strength to it, as well as most of our data is consolidated. And for this, one thing just to point out, so our 4 stars of significance is not comparing wild type to knockout. For each one of these where I ran it, I also used my data to generate a random data set, of surrogate that it was compared to. So this significance is how certain I am that there was phase amplitude happening or phase amplitude coupling happening in these samples. And then from that point, I used a formula just to generate a modulation index, which is a way of comparing it. So now I can compare my wild type to my knockout. And for this system, a lower modulation index shows stronger pack. And as we can see, our wild type shows much stronger phase amplitude coupling, both in our awake state and our non-REM state. And we can see that here, and this is measuring that red bar. Our red bar is reduced. And this angle, what we're seeing is where is that red bar pointing? And it's interesting that it shifted about 90 degrees, which if you remember, that's really what we were talking about with phase amplitude coupling being out of sync, is we're not reducing or altering either of the waveforms, we're just pushing them into different time zones, essentially, just so they're not syncing together. So this shows that these Ift88 knockout mice actually have reduced phase amplitude coupling, which is going to heavily impact their ability to form memories. And this was done using the Canalty method, which is kind of the gold standard. He really came up with the statistics behind measuring phase amplitude coupling, but it is a slightly older method. I also used an alternative method, which it's a little more statistically heavy, but it better corrects for oscillatory power and a few other errors. So it's a little more modern way, but sometimes people don't like really heavy statistics and filtering done on data, so I included both. And what's cool is we see the same results. So in this case, important to note, it's now opposite. A higher pack score shows stronger pack. But again, we get the same story in the same picture. Our wild type mice have much stronger phase amplitude coupling than our cilia ablated mice, both when they're awake and also in non-REM sleep. And this is just showing, so this is where the peak activity was, both for our slow waveforms and our fast waveforms. It's just showing they weren't heavily impacted. We can better see that actually here on this graph. So this is just a heat map of our phase amplitude coupling, and our black tick is just our cursor for where peak pack was. And again, we're just learning that it hasn't moved anywhere and the individual wavelengths aren't reduced. It's just that they're out of sync. So in conclusion for this, there's really kind of two cool takeaways, and they apply to two different areas. The first thing is that I saw reduced EEG waveform activity across a broad spectrum. And that's important because it's a very good system for what we see in depression patients. So we're planning on teasing this out and looking into it a little more. Because again, we want to start working our way backwards. We've got our human evidence, now we've got growing mouse evidence, and then we'll take it one step down more onto the cellular level. And then the second big thing is that the ablation of Ift88, which therefore ablates primary cilia, attenuates our phase amplitude coupling, which is a very important process for memory. It is impacted in a number of neurological diseases and conditions. So on that note, I would just like to thank everyone for the Chen Lab, my old laboratory up at UNH. Mao Chen was very instrumental with helping me with all of this. But I'd also like to take a second to thank Aldebaran for taking me on in this new lab and giving me somewhere to grow.
Very nice, Matt.
And that's my the end of that chapter for me for UNH.
So I'll jump in and say that those were both great, and I'm going to open this for questions if anybody has questions for either of the speakers.
If no one else is going to jump in, I'll jump in. Can I ask something of the first speaker?
Certainly.
You gave a very nice presentation about the movement of smoothened. When you look at that movement, is it that the receptor moves first and then the G protein is already resident within the cilium, or is the translocation of the GI protein as well? In other words, is there essentially coupling between receptor and G protein within the cell body and then migration, or is the migration and what's the sequence?
So to my knowledge, when smoothened is translocating from the, it's a kind of complicated translocation too. There might be a sort of vesicular component to it. When it's translocating, to my knowledge, it's not dragging with it the G alpha I receptor, G alpha I subunit.
Okay, no, it's just
so
it's
what
during
we're
the.
looking at is when I'm using the mini G constructs, I'm looking at something that's probing the activated smoothened receptor.
Yeah, I picked up on the fact that you said that it was over about a 10-minute period that the mini gene was recruited into the cilium.
Correct, but you can watch it, and within minutes you can start. I should have shown movies, but you can, if you look at a time course, you will see it starting to happen, within minutes, and then just over about 10 minutes, it's accumulated quite robustly next to that activated receptor.
No, thank you very much.
The receptor is also going there. So, okay. But I see what your point is. Yeah.
It is obviously not for a calcium audience necessarily, but for people interested in the initial steps of the signal transduction process. It is
Yeah,
interesting.
That's actually a really good question.
Okay,
Might be some
thanks.
way to figure that out. So with fret between the two or something like this. But it could be, or bleaching. I think it may be a photo bleaching experiment might help. Yeah. Okay, thank you.
So I have a question for Matt. Matt, can you distinguish among the different types of memory? So, for example, prefrontal cortex would be more executive functions, and then there'd be short and long-term memory. Can you think you'll be able to do those kind of distinctions?
So we could with more specific behavioral and memory tests. Because as I said, with my experiments and what I was showing, it was just a continuous 24-hour spontaneous activity. If we repeated these but did different specific memory paradigms, we could start to tease out more which areas are impacted. Additionally, with the not the chip, but the intracranial implant that I used, it really only mounts one way, and I am somewhat limited to those. But there are different chips and devices that we could use, again, to better target different brain regions, which we could couple with different memory paradigms to start looking at how different impact or different forms of memory are impacted.
Yeah, because I was impressed that you had only 3 locations that you were measuring from, and you got quite a lot of interesting information, but some of the regions might not be exactly where I would have located. I might have.
Yeah, and so because part of this was, this is all really novel research and going into a new area. So my advisor didn't want me to attack it too specifically and not get a result and then us be discouraged and not evaluate it. So we wanted to be more broad with our experiments just to see if anything's happening and then start teasing in. As I said, Ift88 is a total knockdown of cilia compared to just AC3 is really what we're interested in. And then yes, that intracranial transplant is also rather vague compared to being more specific. So this is kind of like our good preliminary starting point that he can then, he can take, not me, because I'm off. But he can start to tease out and go into more specific directions.
I see someone has raised their hand. You should just go ahead and ask your question.
Hi, my question is to Aldebaran Hofer. So I see that I can see in your slide that there is no STIM1 in the cilium. So I believe that ORI1 is not the predominant pathway for the calcium influx. So what are the predominant mechanisms that influx take place in the cilium?
So, well, there's the famous one would be the polycystin 2, as Barbara knows very much about this. So they're mostly non-selective TRP type channels that are present, but it kind of also wouldn't make sense for the cilium to have STIM and ORI because there's no ER there, so we can't have that coupling, but I suppose it's possible that there could be ORI there somehow, but to my knowledge, there isn't.
Is it possible that just calcium diffuse from the other part to the cilium quickly?
Yeah, it could. Yeah, you mean from the base to the tip?
Yes.
Yes. Yeah. In fact, the reason I did that experiment was I was wondering if there was any, very interesting clustering of STIM and ORI at the base that might favor some kind of signaling into the cilium, but I couldn't really see any evidence for that. But,
thank you.
Yeah.
Go ahead, Sandeep.
Oh, hi, Sandeep.
Hi, how are you?
Good.
Great talks, beautiful imaging, and just it must be really difficult working with these tiny structures, especially when they're wiggling around. So that's a real technical tour de force there. General question: you mentioned this calcium gradient at the beginning, between in the cilia and the cell body,
Right.
and these various channels that are presumably leaking. But to me, if there's no barrier for calcium, right? Because that cutoff for the pores was pretty big, right? 40 kilodaltons, I think you said.
Right.
So does that mean these channels are like, you know, really fluxing calcium in order to maintain that gradient? Especially if it's like 600 nanomolar, right? I think you said it was in the cilia versus, let's say,
that was one of his original. That was David Clapham's work, of course. But
I mean, what are your thoughts on that? Have you
my thoughts are that you've got this compartment that doesn't have all the normal buffers that the cell body does. It doesn't have mitochondria. It doesn't have ER to reuptake calcium. It doesn't have, as far as I know, PMCAs or exchanger, sodium calcium exchanger to control it. So I think if the cell body had none of those things available to it, you would also have just sort of probably an elevated calcium concentration. That's my feeling.
But wouldn't the rate of diffusion just let it dissipate almost immediately?
But it's continuous.
But again, that suggests there must be quite a lot of flux. I don't know. I guess if you've got no extrusion mechanisms.
The volume's really tiny too. You got to think about that. And just its geometry and the volume. It's got a big surface to volume ratio. It's the other aspect of it.
Yeah. Okay. Yeah. Thanks.
You're welcome. Oh, Garrett.
Hi, Aldebaran. Sorry, I don't have my camera on, but great talk and really educational. Maybe you mentioned it, but it was really nice with the EGTA. You showed the calcium going down in the cilia. Like if you take out the calcium on the outside, did you apply such protocols? Let's say where you were looking to cyclic AMP levels, or maybe I missed it and you. Because I thought you had an inhibitory arrow at some point, but then maybe that there wasn't this interplay, and I was wondering on what was that based.
Yeah, so we probably need to revisit this, but one thing that was kind of surprising to us when we removed calcium from the outside of the cilium, there wasn't any effect on the resting cyclic AMP in the cilium. However, we did have some experiments where we put very high calcium outside. So we had like 5 millimolar calcium outside, and that actually increased cyclic AMP or increased the fret ratio in the cilium. So I don't know what is the meaning of that. That's something kind of worth investigating. It could be a calcium activated cyclase, which has also been shown to exist in cilia. Yeah.
Okay. And a very naive question to Matt, also a very great talk, great analysis. Did somebody look to, let's say, classical depression models, and is there, let's say, chronic restraint or the glucocorticoid models, and is there implications in loss of cilia in neurons? But yeah, maybe your research is so forefront, nobody might have looked at that. So, but yeah, it was a consideration.
Just in general, there's a number of growing areas of interest for primary cilia with neuronal function, mainly because of the number of GPCRs that are exclusive to them, and as well as localized there. I couldn't totally hear your question, though. The second half kind of cut
sorry.
out.
Yeah, it was more whether somebody had looked into, let's say, classical, let's say, animal models for depression. Instead of, you look to a knockout, and let's say they develop this depression like feature. I was wondering whether the opposite also happened in, let's say, mouse models for depression, like chronic restraint or other models, whether there have been implication of loss of cilia in those cases in the neurons. So the other way, the arrow in the other way.
Oh, okay, I see what you're saying. In other mouse models, looking at if there has been a loss of cilia.
Yes, you look to, the loss of cilia, and then what does it develop? And coming to depression, I was wondering whether in depression models, loss of cilia have been implicated. Maybe a naive question.
So, no, I understand the question. It's a good question. As far as I know, I haven't seen too many papers where they take other depression models for mice and have looked at the primary cilia. But there have been a number of other neurological papers. such as like an Alzheimer's model for mice, where they looked at the primary cilia and found they were impacted. So for depression, there hasn't been a lot because it's a very heterogeneous disease with a number of different models looking at it. But in some of the other neurological conditions, yes, I do know they've taken a mouse model and then looked at the cilia and found they've been impacted. In the case of Alzheimer's, they even were able to reverse some of the cognitive defects by altering the serotonin signaling in those mice.
I see. Okay. Yeah. Very interesting. Thank you. Great work.
So I wonder whether I can ask Aldebaran. So how is bioenergetics of the cilium working?
Wow, that's a great question. Really good question. Yeah, I suppose they're completely dependent on what happens in the, well, and actually, are they? Great question. So totally open field to my knowledge. But you would imagine that ATP is just diffusing in and powering whatever needs to be done there, phosphorylation, all the biochemistry that's going on.
So you don't think that there is kind of intrinsic glycolytic supply in the cilium?
I don't know. That's, I mean, we could target some ATP sensors there and look at that.
And I guess what is known about the pumps, so kind of usage of ATP in the cilium.
Don't know.
So this
Don't know at
independent,
all.
this kind of independent membrane potential and different membrane potential from the body presumably kind of suggests that there is, well, at least this gives possibility that there is something relevant.
Yeah, I mean, there are also different conductances there. So this could also explain it. But that's another thing that, well, actually I wanted to do is measure membrane potential in that compartment. So I think that would be, could be illuminating.
Okay, good luck.
Yeah,
So
thank you.
So the plasma membrane calcium pump in the cilia is, I mean, there's no ER calcium pump, but is there a exchanger or a?
So I've never, so I'm always poring over these proteomic databases that people publish, and because there have been a number of them, people have done these studies of the ciliome, and I've never seen any pump appearing for calcium, sodium, nothing. So
no exchangers either.
Fascinating.
No, not to my knowledge, but you know, a lot of things don't show up in those screens. So things that we know are there sometimes are not coming up for various reasons.
Interesting.
Okay, great. Thank you very much.
Thank you.
Just going back. Sorry, just going back to the membrane potential thing. They'd have to be electrically insulated for there to be a difference in the membrane potential in the cilia versus the rest of the cell, right?
Yeah, apparently the ciliary necklace is providing some of that. And then also the geometry that you have this long, very long tube, very thin, is somehow connected to this. I mean, I think David Clapham was more the authority on this topic.
But you told us the receptors go in and out, right? So,
right.
And if it's non-vesicular, you mentioned it might be vesicular, but if it's diffusion through the membrane, again, that would suggest there's some sort of continuity, right?
There is continuity. I mean, it's kind of mysterious how you can get the receptor trafficking thing is sort of a mystery to me. So some of it is vesicular, and some of them though do go through the membrane, and they have to traverse that barrier.
Right. Okay. Yeah. Very interesting.
Thank you. I have a question for Matt, if I may trouble him.
Yeah.
So did the Ift88 knockout mice, did they have other phenotypes like cystic kidneys or?
Cystic kidneys we haven't seen. And the other thing is with these Ift88 knockout mice, we had developed them and we didn't do a conventional knockout. These are all induced conditional knockouts. So they're it's a Cre recombinase linked knockout, where we induce it with tamoxifen when they're already 2 months old is when we start it. And then we haven't held on to the mice long enough to see if it further develops. So no, we haven't, like I said, we kind of started off on developing this with the inducible, and we haven't seen too many other phenotypes, but we also haven't too heavily investigated them.
But that's why you don't see situs inversus, because you induce it late.
Yeah. And that's just one of the many conditions that you would see. Because one reason too is, for instance, for our AC3 mice, when you knock out adenyl cyclase conventionally, most of all the pups die immediately after birth. And actually with most cilia mutations and mouse lines that I know of, a lot of times you can get that very either neonatal death or soon after. Because again, they're so important with early development that often any mutation can affect them.
And which mice got really fat? Was it the IFT?
So that both. Our adenyl cyclase 3 mice is the data I showed, but we do see obesity in the IFT mice as well.
And but that must come on pretty early because you don't keep the mice that long.
Yeah, I did not include the figure from that paper on the weight gain, but it is fairly rapid. I'm trying to think of the length of it, but I don't think it's more than 4 or 5 months that he had done that study over.
Interesting.
Can I ask? Very nice talks. Matthew, very interesting. But can you tell us, you might have said something in the beginning, which I missed, something more about this Ift88? What is it doing? What kind of protein?
Yeah, so let me just share that again. And I will go back to it. Yeah, we'll go to this slide. So the primary cilia is isolated from the rest of the cell body by this transition zone, which filters out anything roughly over 40 kilodaltons. And anything larger than that has to be actively transported through. And we can see here, we've got this kinesin molecule in this cargo molecule, which the cargo is essentially a vesicle. Ift88 lines the exterior of that cargo vesicle and tags and identifies it. So it both helps with it moving through the transition zone as well as being attached, primarily with being attached to this kinesin. So when Ift88 is removed, you basically have no way to carry all your building materials to up the primary cilia. Because one thing too that's kind of interesting is primary cilia are not static. They're always elongating and always retracting. They're just in an equilibrium. So a lot of things involved with ciliogenesis and retraction are often based off of just disrupting that equilibrium.
Is it a conserved protein? When you compare different organisms?
Yeah, so a lot of the cilia, I'm trying to think if there's more specific, but yeah, a number of IFTs as well as general cilia proteins are often well conserved across a number of organisms. I know at one point I had actually done a little evolutionary biology project on that, and we did see that a number of them are well conserved. And there are a lot of them, as we see here, Ift88. So there are varieties and differences, but across species, they are a well-conserved protein.
Thank you.
So Barbara, we should probably, Martin here, so we should probably thank Aldebaran and Matt for two actually really striking talks. I mean, lovely images and, you know, really amazing analysis, Matt. Really super interesting. It's always great to learn something new, and I learned a lot new today. and actually a really, really good discussion too. Unless anybody has a pressing question, and everyone's more than welcome to stay, I suggest perhaps we let our speakers go and get a glass of water, whatever it is they might need, and just thank them very much, and also to you, Barbara, as well, and congratulations again on your 70th. For those folks that haven't heard, although I think everyone in calcium signaling knows.
You'll send out announcements for the next, because thank you for reinstating these webinars, and